So all of the images that we're going to look at here were submitted in lab reports from students who have taken this class before. So all of these are student-generated images. So first we've got some images of some cells. And so over here are some unstained cheek cells. And so what we see is a human cell, cheek cell with a nucleus, another cell with the nucleus in the middle, and this cell, and you really can't see the nucleus so well. But what you see are individual cells. In some of them, we can see the nucleus. That's great. Perfect. Over here for the stained cheek cells, we see from another student that essentially we've got a tissue mass. You can see the stained nuclei here, right? And then here's a cell, but you can see there's a whole lot of lines in here, and that's because you've got a whole bunch of cell on top of another cell, on top of another cell, on top of another cell. So while that's interesting, even with the stained ones, I would like to see individual cells if possible. Now here's an example of, this is low power stained cheek cell. And so you can see this image right here, the person actually was using their phone camera up against it, and we're able to get some decent images. So here are the stained cheek cells that are these bright yellow. These are at the low power, and at the high power we can see that these images, and this image it's pretty good. Compare it to the one before where we can see them really nice and clear. This is really blurry. That's not good. This is pretty good, right? I would take this, and these are just not very good at all. Now all of these other dots and splotches, those are either debris that are on the slide itself, or they're actually in the optics of your microscope. For example, if you've got a bunch of dust on your ocular, then what can happen is you will end up seeing that. So, one thing that students will do is sometimes they'll focus in on this thing thinking that that's correct, when really this is the thing to focus in. So always know what you're looking for, right? So, and then here is a scanning lens with unstained cheek cells. And the contrast isn't great, but I can still see a cell here, right, with the nucleus in the middle, nucleus in the middle, nucleus in the middle. So here's 3 cells right next to each other. Here's a single cell. So this is pretty good for a scanning lens as well, as long as I can see it. One way to change the contrast would be to change the iris diaphragm. So let's move into some stained mouth samples. And so this, for example, these are stained, right, with crystal violet human cells, dark nuclei in the middle of each one. It's not very good. I can't really see a whole lot of what's going on here, right? I can see that there are cells. So basically I can see that you did the staining properly and that you got it from your mouth. And so, you would get the majority of the points for that, but not all for this, because I just can't see the individual bacteria, for example. Here's one where the image is fairly decent. Here's one human cell with the nucleus in the middle. Here's another human cell. We can't see the nucleus, but all these dots all over are probably microbes that are studded on it. Now, when you see a bunch of filaments like this, and it came from something that there was a swab involved, sometimes that can be the actual fibers from the swab. And if we look at this one up close, we can see more fibers, right? And then here's a human cell, and in the middle is a nucleus. So it's pretty good. It's better than this over here, but compare, these images up here to this one down here. This is beautiful. I can see there's one human cell here, there's a nucleus in the middle here, and then if we look at these little dots here, and I went ahead and magnified them, right, these are individual bacteria. So that is an excellent image. Here's another one from another student who, same thing. Now there's clusters of cells here, but the key thing is I want to see a cell, I want to see a nucleus, and I want to see the bacterial spots all over it. So those are examples of excellent images. This is okay, this is okay. We would need a higher image of this as well. But for lower magnification, that's decent. So now let's look at some stained bacterial samples, right? So for example, from weeks 2 and 3, you saved out a broth sample and a plate sample, and you smeared and heat fixed, and now you're gram staining this week. Now, if we look at this, this is showing essentially this whole mesh network. This right here is probably an individual bacterial cell, and then one here, and one here, one here, but these are all too small. When you're taking the bacterial images, always use 100x oil immersion lens, so I'm not sure why these weren't a little bit bigger. Okay, so this student may have used a lower magnification on this one and just submitted it incorrectly, but we want to use the 100x oil immersion lens. So this right here is fair as well. Here though, we can see individual, there's a cell, there's a cell, here's a cell, here's a cell, and then we've got a whole bunch of cells clumped together. That's what we see here. We can see that it's just kind of fuzzy or blurry. And same thing down here, these are definitely individual cells. So this one, for example, is better, right, than the one over here. It's a little bit better than the one up here because we can see individual cells. This is over here on the lower left. It reminds me of the one on the right, even though it's from a different student. And here we see individual cells, we see clumps of cells, and then this also is probably a long bacillus cell. So long bacillus, and then down here we've got coccus or coccobacillus, and this is another bacillus right here. So these are fair, right? Those are passable. If you get images like that and that's the best you can get, submit it and you will certainly get points, and you should be, not unhappy with them. However, this is better. Right, so what we can see is we've got nice contrast. We've got some individual cells, right? So these are bacillus. This right here, we've got a bunch of individual cells as well. These look like they're coccobacillus, right? They're still all jammed up against each other, but at least we can make out individual cells. For example, down here, this is so meshed up that we can't really see individual cells at all, right? So these are good. And then for really good cells, this is what you're looking for. This really excites me. I'm so happy when students can produce high quality images like this. So look at this. I mean, this right here is a bacterial cell. This is another one. So this would be diplobacillus. This would be just bacillus. When we've got a chain of them, that would be streptobacillus. So these individual cell, individual cell, individual cell, etc. Here's another one. The contrast isn't as good. The student could have used a little bit more light on this, but still, this is a great image. Here are individual bacillus cells all mixed in. Right over here we've got coccus or coccobacillus, and I can see a whole bunch of individual cells. So this is wonderful. What a wonderful image, right? And they're next to each other, but that's okay. And sometimes this is a gram stain. Sometimes what you'll see is that some are purple and some are red, right? You really only want one. You want it to be purple or you want it to be pink. But look, you guys are just starting out, right? I understand that. So if I see something that's not perfect, but the quality of the image is good, you're right on there, and I'm very pleased with this. The other thing is, it depends on how old your culture is. So if your plate or your broth was old, you're going to get this no matter how good you are, right? So I don't worry about that. And now look at this. This is gold, right? Look at that. 1, 2, 3, 4 bacillus cells all together for this streptobacillus. That is such a beautiful image. And this is wonderful as well. This is a streptobacillus. And so we see a cell, a cell, a cell. This looks like it might be a cell that's dividing into two, or right there. So these are chains of bacillus. Okay, so that would be streptobacillus. So these are all just wonderful images. These are the types of images that you're shooting for. Now, sometimes I'll get images that look like this, right? So, just starting out, it's hard for you to know what is real and what is not, but that's where all these images I showed you previously come into play, as well as Google image searches, right? Submitting something like this, you may be like, well, maybe these are bacteria. Well, since you've got all this weird color and contrast, that's just some weird staining artifact that's no good. This right here was just some piece of debris stuck on the slide, no good. This, just some kind of garbage, no good. None of this is good either. Now you may say, but Dr. Well, that looks like a human cell. Well, it kind of does, but this came from a supposedly pure bacterial sample, so we know that it's not even, there should be no human cells in there, right? So, and then this right here, this was supposed to be a stained slide, and the student submitted this, so there's nothing there. There's just various pieces of garbage, debris, optics. When you're getting used to these microscopes, sometimes it's maddening because you're like, I can't see anything but this. My guess is that the student had this out of focus, and that if the student focused in more, maybe these are actual cells, but it's hard to tell. And same with this. See how these are all splotchy? It's not like they're uniform, right? If we go back a couple slides, you see how these are all kind of uniform. They're all about the same size, same shape, same here, same there. If you have a particularly a pure sample, then you should look at this and be like, oh, that's just like little splotches, different shapes, different sizes, not what we're looking for. And then here's some more examples of really, this is just garbage on people's slides. This is just a smudge of stain that's stuck on there that's not bacteria. This is not bacteria. So avoid images that look like this because they're not doing us any good and you're on the wrong path. I hope this has been helpful. I wish you all well on your microscopy. I hope you enjoy it. Remember, patience is a virtue, and I really look forward to seeing images, particularly of your cheek cells and of your bacteria cells, posted in the discussion board, or if you don't do that and you do fruit fly instead, that's fine. But when it gets to the point where I see your lab report, I'm really looking forward to some good images. Hopefully something like this. If not, then perhaps something like this, right? Or like this, but definitely not any of this stuff right here. All right, and with that, good luck.